Hi guys, my name is Dan. I am, I was in the experimental surgery master's program at McGill, and as part of my master's, I did a thesis with Dr. Del Maestro as my supervisor. I did my thesis at the Neurosurgical Simulation and Artificial Intelligence Learning Center, with Dr. Del Maestro being my supervisor. And my thesis, which took roughly a year and a half to fully finish, was entitled Quantitation of Tissue Resection Utilizing a Brain Tumor Model and 7 Tesla MR Imaging Technology. So it's a very long title, but during the presentation, I will break down what each of the components means. Perfect. I wanted to give you guys a bit of a background as to why we did this study in the first place. Neurosurgical residency training is one of the most demanding in the realm of surgery because trainees are required to master a wide range of psychomotor skills. First, by manual dexterity, then the ability to work at a microscopic level, and finally, the ability to apply force in a way that does not damage fragile tissue. Here we're talking about brain tissue like pia or gray-white matter, etc. So working at a microscopic level, the magnitude of force we apply is totally different. In parallel to that, I wanted to highlight the fact that in the last decades, there has been a departure from the graduated responsibility model in the way trainees are being trained. That means that instead of giving them more responsibilities as they advance in their training, in fact, they are exposed to less opportunities in the operating room. Given the fact that roughly a quarter of complications in neurosurgery result from technical errors, and that of all surgeons facing litigation in courts, 34% are neurosurgeons, there's a clear need to find new ways to give training opportunities to trainees. And to do that at the Neurosurgical Learning, at the Neurosurgical Simulation and Artificial Intelligence Learning Center, a machine has been developed. Everyone knows it as the NeuroVR. It's a neurosurgery simulator that allows users to train on different levels of difficulty. It offers auditory, visual, and haptic feedback, and it is essentially, from the picture as you can see, a whole setup with a computer where the user can see a brain in virtual reality through goggles, and has 2 tools that mimic the same ones as surgeons use, and that allows them to essentially operate on the brain tumor. Now, this machine is all it's a very good tool as we can see it here, but it needs, just like any other tool, validation, and we have reached a stage where we need to give it predictive validation, meaning to prove its ability to determine the level of expertise of a user. To do that, one of the members of the lab, Dr. Winkler-Schwartz, came up with a framework for predictive validation. It's a very elegant flow where the idea is to take neurosurgical residents and to have them trained and perform on an animal tumor, meaning an animal model that contains an artificial tumor. Then, in a randomized controlled trial, these surgical residents are divided into 2 groups, one control group that goes through a standard residency process, and an experimental group that on top of the standard residency receives training on the virtual reality machine, the NeuroVR. After that training is over, both groups are reconvened to the lab, and they will once again perform surgery on the animal tumor. So Dr. Schwartz has already come up with a few aspects of the randomized control, notably in his paper, Machine Learning Identification of Surgical and Operative Factors Associated with Surgical Expertise in Virtual Reality Simulation. In essence, he has identified in this paper the factors that are most important when assessing expertise in the realm of neurosurgery. Now, the next step after that, if I can go back to this slide, was to create an animal tumor model which can help to assess surgical performance. And to do that, a second paper came out this year called Creating a Comprehensive Research Platform for Surgical Technique and Operative Outcome in Primary Brain Tumor Neurosurgery. So this model that was proposed is used to assess one specific technique, a demanding technique called Subpeel Technique. It's used not only in brain tumor resection, but also in epilepsy surgery. This technique offers numerous advantages because what it entails is essentially peeling a bit the pia of the brain in order to remove the tumor in a supertotal fashion. First, by conserving the pia, it allows to protect the normal anatomy of the brain. It also reduces bleeding, and it leads to longer lengths of survival because of the supertotal nature of the resection. By supertotal, we mean that instead of just removing part of the tumor, not only is the entire tumor removed, but also a bit of the healthy layer of brain tissue around it to make sure that there is no resurgence of the tumor after the operation. While this technique is highly efficient, it is very demanding in terms of the technical skills. And to highlight the technicality of it, I wanted to show you this short little video. Oh, sorry. And it is not working. Well.
Do you have it somewhere else on your system?
I can exit the presentation mode to simply show it if I can. Oh, here we go. Perfect. So in this short video, as you can see, Dr. Schwartz with his left hand lifts the peel layer, and with the sonopet starts removing healthy tissue around the tumor. Now, this is not a real tumor. This is a calf brain which has been injected with an artificial tumor. I will explain later on what this artificial tumor is all about. So here, as you can see, he goes under the pia and removes the healthy tissue around the tumor itself. Then comes what we call the tumor debulking in the next stage, where essentially the tumor is removed. It's been exposed now and it is removed. Finally, he cleans the surgical cavity to make sure there's nothing left in there. Okay, so back to the presentation. Okay, so I mentioned that this is an animal brain, and there has been essentially a lot of investigation on our part to determine what physical model, actually not even animal, but what physical model is best to be used in this case. First, out there, the most common models are called full gel models. Essentially, a big block of gel is used, and within that big block of gel, a smaller block of gel of a different consistency and appearance is nested. This sort of mimics a brain with its tumor inside. It offers advantages because it's cheap and it's easy to make, but of course it does not offer any sort of realistic setting. It's not a real brain. So moving in the higher level of realism, we have cadaveric models, meaning using cadavers to perform these tasks. One example are endoscopic endonasal skull-based resection of polymer tumor, meaning that a polymer is injected in the skull of a cadaver, and it's accessed using the endoscopic endonasal resection method. You can also inject resin instead of polymer, and both of these models are very realistic because, of course, you use a real human cadaver. But of course, you can imagine that the cost of it makes this method not very cost-effective. Not every trainee can have access to a cadaver. It would be logistically impossible to do. So the most practical models are animal models because they're cost efficient and they still offer a certain degree of realism. The first ones that we thought of are the porcine head models, meaning pig head models. So a whole pig head is used. Although it is sort of realistic in that it offers gyri and sulci, it is highly inconvenient because the size of the pig's head is the pig's brain is very small. So by the time you remove the skin of the head and you drill through the skull, all that effort yields you only a tiny little brain. So finally, we settled for an ovine models. Ovine meaning sheep, sheep, and bovine meaning beef. Not beef, sorry. What is the animal? Cow model. So In these models, the brain is bigger in size, which is more convenient, and some models have taken the entire head. So the entire head of the cow or the sheep is taken, and an artificial vascularization is plugged in. So tubes with colored dye are injected in the main vessels of the head to recreate the circulatory system. It's highly realistic, but it's not practical because every time you want to operate, you'd have to hook the head on a pump and on a lot of vessels. This is not doable in a lot of settings. So the very last model, and the closest one to ours, offers essentially an artificial tumor in the form of an agar-agar solution with fluorescence that is injected into the brain. Based off that, our model took that and went beyond in terms of its in terms of its realism. So the first element of our model is an ex vivo calf brain. I emphasize ex vivo, meaning that the calf brain is not within the head. We bought the brain outside of the skull. So we just went to the butcher and we got the brain. The calf brain, so meaning a baby cow, is appropriate in our case because it's adequate in size and anatomy and it's cost effective. The second component of the model is the artificial alginate tumor. Alginate is a powder made out of algae, and mixed with water and a cross-linking agent, it can allow the gel to go from liquid to solid simply by resting it at room temperature. It's also very convenient because we can adjust the stiffness of the gel by changing the concentration of calcium sulfate. We can also add other components like gadolinium, which is a component that allows for the tumor to be hyper-intense when we scan it. Hyper-intense meaning that when we scan it in a 7 Tesla MRI, it comes out as if it were glowing, so we can really see it nicely. All of this is very inexpensive to produce. So this brain model has the advantage of the cost, which it can encourage residency programs to use it. Now, this is where my paper comes because so far we've talked about how we created the model, but the next step is how do we study the model and how do we study surgical performance on that model. So this paper, which has been accepted by World Neurosurgery and it's in press, is called Quantitation of Tissue Resection, utilizing a brain tumor model and 7 Tesla MRI technology. If I break down the components of the title, what we're trying to do is, using a brain model, we want to use a 7 Tesla MR scanner in order to assess quantitatively tissue resection. So that's what it means. And the purpose of this study is not to create a full-fledged sort of analysis model, but it's more of to come up with a pilot to prove that we can use the 7 Tesla MR technology with the brain tumor model we came up with in order to quantify and understand complex neurosurgical skills. So to our knowledge, this is the first time both technologies, so the brain tumor and the 7 Tesla MRI technology, it's the first time that both technologies are being used together. Before the study, we set 3 goals that we wanted to address. First, we wanted to see if, using a brain alone with no tumor in it, if we could quantitatively assess healthy tissue removal. Simply put, we took the calf hemispheres and we cut small chunks of healthy tissue, and we wanted to know that based on a 7 Tesla MR scan, if we were able to determine how much has been removed. Afterwards, the second goal based off that is to use this technique to quantify alginate artificial tumor removal. So in the next level of complexity, we inject the tumor and we want to know if we're able to quantify how much tumor has been removed and how much is left. Finally, in the last stage, we wanted to determine our ability to quantify white matter, gray matter, and total tissue resected on brains with alginate tumor. So using the brain with the alginate tumor, we wanted to know that on top of the tumor, if we could quantify how much white matter and gray matter around it has been removed. The last of the components is to perform a holistic visual assessment of damages. So essentially, based on the picture of this on the scan, if we can say how much damage has been done to the brain. Again, this study is a pilot and a proof of concept, so by no means is it a definitive model. So in this study, we used 7 calf hemispheres. It's important that I emphasize hemisphere because we did not use the full brain. We bought full brains, but when we brought them back to the lab, we looked at the 2 hemispheres and we picked the one that we thought was the most, how can I say that, undamaged, the best looking one. So then we separated both hemispheres and only kept one. We divided each hemisphere, the 7 hemispheres, sorry, into 2 groups. Group 1 had 3 hemispheres and group 2 had 4 hemispheres. Group 1 was the group of brains we used to determine our ability to quantify healthy brain tissue removal. So that's the group of brains that were removed healthy chunks of tissue, chunks of healthy tissue, sorry. All of these brains had fiducials, and I'll explain in the next slide what a fiducial is. In group 2, we had 4 brains, and these brains all had tumors. Each brain had a different tumor size. So the tumors went from smallest at 0.5 milliliters to biggest at 1.5 milliliters. Only one brain in this group had fiducial. Now, fiducials are simply 3D printed pieces of plastic. They are in the shape of nails, and the goal is essentially to see their usefulness in improving registration. So let me explain that a bit. These pieces of plastics are inserted on the surface of the brain, and they're used in order to essentially offer landmarks for registration. Registration is the process of taking one scan and superimposing another scan on top of it. So we take the preoperative scan, we take the postoperative scan, and we superimpose them. We found that these fiducials work as very good landmarks for rigid registration. And I'll explain that a little later on in the presentation. Now, why did we pick a 7 Tesla MRI? We put a lot of emphasis on saying 7 Tesla because in clinical settings, most scanners are 1.5 Tesla or 3 Tesla at best. We use a 7 Tesla, which offers us a higher signal-to-noise ratio. As you know, in MRI, the picture is not the clearest, so there's a lot of noise, a lot of interference. It also allows us to have higher resolution for the landmarks, like white matter tracks. And it's very important when we talk about hyperintense tumor. So the tumor, when we scan it, is glowing, which can sort of, affect the visibility of adjacent white matter tracks. By having a 7 Tesla MRI, we want to give higher resolution of those landmarks, which will help us in the process of registration. So the first step is after purchasing the brains, we need to scan them in order to obtain a preoperative scan. So after cutting the brain hemisphere, we take it and we wrap it in gauze. We then soak it in fluorinert, which is a chemical that allows us to reduce the amount of noise and interference, and we insert it in a plastic tube. This plastic tube is chosen so that it fits perfectly the brain. For some brain, it's a little tight. For some brain, it's a little big. But overall, it fits right in the plastic tube. We then send the tube to the scanner, and it takes roughly 24 hours for the scan to be produced. We take that data and we process it using a software called 3D Slicer. The first stage in the process is to filter the noise out of the scan. So you can imagine that the scan is not completely perfect, so we apply what we call an N4 ITK bias field correction filter. It allows us to normalize the signal in homogeneity in each volume. The next thing that we do is called segmentation. Segmentation in computer vision is a process where we divide a digital image in multiple segments. It's simply labeling each segment. So in this study, we used 4 segments. We used a gray matter segment, a white matter segment, a tumor one, and a fiducial segment. The fifth one, which is air here, is removed from the final segmentation, so we don't really count it in our 4 segments. The segmentation process occurs in 2 steps. First, a manual segmentation occurs. What it means is I had to go through 20 to 30 slices and hand draw each structure. So that was very time consuming. I had to make sure to draw each white matter tract finely and make sure that the contour that I drew was exact, that it didn't encompass any structures it should not have encompassed. From that, after I complete roughly 20 to 30 slices manually, I used semi-automatic segmentation. So a different algorithm will apply essentially segmentation across the entire volume of brain. For the tumor, I used something called Otsu's thresholding. And for the gray and white matter segments, I used seeded region growing. So from a seed, from a segment, it expands throughout the entire volume across different slices. After that is done, I smooth the image. It's a lot of technical terms, but I think the image will better describe what the process is. On the left, you have the image as it comes to me after biased filtering. So this image has been filtered, but it's not segmented. And on the right, you have the segmentation, the segmented image. So you can see on the right-hand side the white matter tract in white, and you can see the gray matter in green and the tumor in yellow. You can see that the segmentation offers a tiny little bit of pixelization, but that's normal because the MRI technology can't allow us to have something super smooth. So this is the best that we can come up with in terms of smoothness. Once we have these images of the preoperative brain, we do the same with the postoperative scan. So the scan of the brain after the tumor has been removed. And once we have those 2 segmented brains, the most important step comes in, which is registration. Registration, as a reminder, is the process of superimposing 2 images. So again, we used 3D Slicer, and we used a specific extension called segmentation registration. Registration occurs in 2 steps. First, a rigid registration, and then an affine registration. Rigid simply means that the 2 images are superimposed. Affine means that not only are they superimposed, but one image is slightly distorted. So it's either compressed or stretched so that it fits on the other image. And the result is as follows. Here I color-coded them so we can see a bit clearer, but the white matter tracks here have been superimposed, and it's not a perfect fit, but it's close enough to give us a lot of information. The most crucial area of superimposition is right here. That's where the tumor is. So this is the preoperative tumor, and these 2 branches are from the postoperative one. And it gives us a lot of information on what has been done here. This sort of information is not available to the naked eye, so we can only see it through a scan. Moving on to the results section, the first goal, as a reminder, was to quantitatively assess healthy brain tissue removal. This is what a hemisphere looks like before a chunk of brain is removed, and this is what it looks like afterwards. So here you can see 2 little holes, and these are where chunks have been removed. On the scan, it looks like this. This is a coronal slice with a scale bar of 25 millimeters, and this is the postoperative scan. So you can see here, there's a chunk of brain missing. That afterwards is segmented. The 2 images are superimposed, and we subtract the 2 images to get this, what's been removed. From that, we're essentially able to create this table where, based off the scans, we can determine the exact volume of tissue removed. This is the weight in grams that we recorded upon removing the brain tissue. The volume here is obtained from the registration process. So the software returns to me, in the case of brain 1, the value of 0.214, 0.696, 0.821 milliliters. You will notice that for two chunks, chunk number 1 and chunk number 3 on brain 1, we were not able to get any values, and that's because the brains in these areas have been distorted so much that it was impossible to bring it back together and do the registration. Nonetheless, if we do a percent error analysis, we're able to essentially get the volume and the predicted weight down to a margin of 0.11% to 7.6% error. So it's pretty decent. It's not perfect, but within the bar of 10%, we're not hoping for that in the beginning. This is a plot of the recorded weight versus the volume, and from there you can see that there's almost a linear relation between the two, which allows us to get a polynomial. We found that the correlation coefficient was more than 0.99, and so was the coefficient of determination. Of course, we're talking only about 4 points here, so it's certainly not enough, but it's a good beginning. It offers us the confidence that if we were to add more points, the relation would be likely linear as well. So there were some limitations with this approach. The reason why we were not able to get two of the chunks was when we put the brain in the cylinder, we compress it a bit. So it deforms the brain a lot, which gives us a harder time at the registration process. In addition, it's not the entire brain that is scanned, but only a specific chunk of it. That is inherent to the scanning method. We can't do anything about it. So every time we have to make sure that whatever we want to scan falls between that window. Unfortunately for brain 1, although we were able to scan it initially, after all the processing, somehow those two chunks found themselves outside of our window of view. It's unfortunate, but it at least gives us the information that for the next time, we have to make sure that whatever chunk we remove falls perfectly in the middle of that field of view. Finally, and this is a bigger weakness, the smaller pieces of tissue resection are hard to detect. So there is a certain amount of resection below which we can't detect any difference. So it can be easily mistaken for simply brain surface deformation instead of actual resection. So after this first part, we were confident in our ability to determine the amount of tumor removed. So moving on to part 2, which is the quantitative assessment of tumor resection, we applied the exact same processing to brains that had tumors. So in figure A, this is brain 1 of group 2, and you can see the tumor here within our region of interest, the rectangle box. These 3 views are zoomed views of this same brain. This is an area of the pre-resection brain that had no tumor. This is also the pre-resection brain, but with the tumor. And this is the post-operative brain with whatever remainder of the tumor there is. Now, you can notice these little letters, NG, NW, NG prime, and NT prime, NW prime. These are quantities that we defined to label preoperative gray matter, preoperative white matter, preoperative tumor matter, and postoperative gray, postoperative white, and postoperative tumor. Based on these quantities, we used a set of 7 equations to determine an array of quantities. So if I wanted to know, for instance, how much matter there was within my region of interest, all I needed was these 4 pieces of information. Essentially, when we do the segmentation process, we're only able to get some of the information. But using the information available, we can get the remainder of the quantities. So I can get the difference in gray matter, the difference in white matter, the difference in tumor, etc. So using this set of 7 equations, we were able to first determine how much tumor was in the brain at first and how much has been removed. It's very interesting here because the volumes that you have here are the volumes that we determined upon injection. So when we create our mix, we then plug in the syringe, and we see, okay, based on the syringe measure, there is 0.5 milliliter of alginate tumor. But then when we do our segmentation, we realize that, well, there's not quite 0.5 milliliters, but there's more 0.421 milliliters. And it gives us an information, which is that the mixing process is not necessarily highly accurate. So you can see here that overall, there's not that big of a difference except for brain 2. So you have these milliliter values for the volume of a tumor that we determined. Then we were able to determine the length of the tumor. And this factor is quite important because the length of the tumor sort of correlates with the volume of tumor injected. We found that the longer the tumor was, the more volume we injected, the longer the tumor got. And this will affect our ability to capture the entire tumor upon scanning. Using our segmentation method, we were then able to determine how much tumor was left. What you have to realize is when we use the sonopet during the surgery and we remove the tumor, we don't really know how much we're removing. Unless you remove everything, then you will know, of course, how much you have removed. But if you do a partial removal, as in brain 1 here, there's no real efficient way of knowing how much has been removed. Now, using our scanning method, we're able to get that value. We're able to determine that out of the 0.421 milliliters of tumor present, we were able to remove 0.238 milliliters, which represents 56.5% of the total volume. And it means that there's a residual 43.5% of tumor. This sort of information is invaluable because it will tell us if a surgery has been fully completed and if it's been successful, etc. So of course, in this case, whoever left that much tumor left quite a lot. There's almost half of the tumor left in there. Moving on to our last goal, now that we know how much tumor has been removed, we also want to know how much white tumor, gray matter, and total tissue has been removed. So in our case, we used sort of the same approach. So we superimpose gray matter together, white matter together, and we subtract. And we're able to get this table where we first see how much gray matter has been removed, how much white matter has been removed, and then we compute an important ratio, which is the ratio of gray matter to white matter resected. Now, let me explain a bit. If we go back to what the Subpeel technique is, it's supposed to lead to a supratotal resection, meaning a resection of the tumor and of the healthy matter around it. So normally, what happens is we should have more gray matter than white matter resected, because this is simply based on brain anatomy. We are supposed to remove an entire gyrus. A gyrus is made mostly out of gray matter, so that ratio should be pretty high. It's interesting to note that in brain 1, where the tumor was incomplete, the ratio was kind of close to 1, whereas in brain 3, where the removal was actually supratotal, the ratio is 11:1, which clearly here the entire gyrus has been removed. This sort of information, again, can be derived from the scans, and it gives a lot of information as to how successful the entire surgery was. Finally, the last step was to perform a holistic visual assessment. So it's good to have all these quantitative measures, but we want to know, based on a naked eye assessment, if there's been any damage to specific structures. And here you can see that, for example, there's a tumor left, but there's a whole section of a white matter tract that is missing. Ideally, only this would have been removed, this gyrus, but here there's an entire piece of white matter being removed. So this sort of information is also very useful based on the scans, and it essentially tells us that a scan can tell us quantitative information and qualitative information. Again here, another example. this is a successful tumor resection, because we see here the tumor has been fully removed, and along with it, a significant portion of the gyrus has been removed too. So I wanted to show you a small video. This video shows scrolling through pre and post resection segmented images of brains with allogeneic tumor. First, we will look at brain 1 of group 2 to show subtotal tumor resection. From the post-resection image on the right, we see significant amount of tumor left after the resection. In addition, white tumor tracts around the tumor have been damaged. Therefore, this operation did not meet our criteria for success. We will now look at brain 3 of group 2 to show complete tumor resection. No residual tumor is left, and most of the gyrus containing the tumor has been removed as instructed. However, damage to healthy tissue in the surgical wound indicates excessive force application with the surgical tools from the operator. Okay, so that last part, the excessive force application, it's not a definitive assessment. It's a bit speculative in that we look at the scan, and if we see that there has been significant holes, unnecessary holes outside of the removal of the gyrus, we can conclude that at that point, a little bit too much force has been applied. So there are of course limitations to our approach, and all the issues are essentially related to the registration process, because regardless of how accurate our segmentation is, if we can't put one image on top of the other, there's no analysis possible. We found that first, we need the surgical wound to be kept open, meaning that if upon putting the brain into the tube, the hole collapses onto itself, there's no way we can register, because there's no way we can reopen that hole and make it wrap around the tumor on the preoperative image. So a solution that we propose is to inject a non-compressible substance that solidifies at room temperature, and that is not hyper-intense. It does not show on the scan. The second limitation that we found is the entire brain has to be scanned. If we miss some structures, registration is impossible. The third point is that brain compression due to the plastic tube causes deformation. So some brains are slightly bigger than others. So when we push them in, they are compressed, and it therefore deforms landmarks. It can leave certain areas out of the field of view, and it's harder to register the deformed landmarks. And finally, if the tumor is too big, it starts pushing on adjacent healthy tissue, and it deforms the landmarks. So the tumor can only be of a certain size. It can't be bigger than that, which is a limitation, because of course, trainees won't be exposed to cases where the tumors are larger. This brings us to the conclusion of the study, where we fulfilled our 3 goals. So I will not repeat them, but we essentially fulfilled them. And it increases the granularity of the data obtainable from operative procedures. We give more information on top of the simple naked eye assessment upon surgery. And it opens the door for future studies and skills transfer between virtual reality simulators and the operating room. Both can be complementary, and both can work hand in hand to give trainees more opportunities outside of operating rooms. So the distant goal of this study is, of course, the validation trial that I mentioned at the beginning. But before that can be fulfilled, there's one thing that has to be done. Remember how I told you that the segmentation process starts with manual segmentation? That is way too time consuming. So the only way this whole thing moves forward is if an automatic pipeline for segmentation and registration is created. Essentially an entire algorithm where I only feed the algorithm with my 2 pictures, and it tells me everything after it's being processed. Of course, that's going to take time to do, but it's very possible. It's already possible in human clinical studies. So it's only a matter of investing time into doing that for calf brains. This is the list of people who collaborated on the project. Dr. Winkler-Schwartz. We collaborated with Dr. Collins and Dr. Giziri, with Dr. Rutko and Marius Tuznik for the scanning. At the moment in the lab, there were Aiden, Rikai, Dr. Karlik, Summer, Swajan, and myself. We also benefited from the help of Dr. Adrian Del Maestro and from former graduates, Nikan, Nicole, and Vincent. So yes, thank you very much for listening to this presentation.